Some cells are programmed to die. Apoptosis is programmed cell death, a series of events caused by the expression of certain genes. Many of these, death genes, have been identified, and related ones have been found in organisms as diverse as nematodes and humans. Apoptosis is vital to the normal development of all animals. For example, the nematode C. elegans produces precisely 1090 somatic cells as it develops from a fertilized egg to an adult, see figure 10. But 131 of these cells die. The sequential expression of two genes, called CED4 and CED3, for cell death appears to control this process. In this system, for example, there are 302 nerve cells that come from 405 precursors, thus 103 cells undergo apoptosis. If the protein encoded by either CED3 or CED4 is non-functional, all 405 cells form nerve cells, and disorganization results. A gene, CED9, codes for an inhibitor of apoptosis, that is, its protein blocks the function of the CED4 gene. So, where cell death is required, CED3 and CED4 are active and CED9 is inactive. Where cell death does not occur, the reverse is true. A similar system of cell death genes acts in humans. During early development, human hands and feet look like tiny paddles. The fingers and toes are linked by connective tissue. Between days 41 and 56 of development, the cells between the digits die, freeing the individual fingers and toes, figure 11. The protein, an enzyme called caspase, that stimulates this apoptosis is similar in amino acid sequence to the Institute for Advanced Studies, Jodhpur 10. Protein coded by CED3, and a human protein BCL2 that inhibits apoptosis is similar to CED9. So humans and nematodes, two creatures separated by more than 600 million years of evolutionary time, have similar genes controlling programmed cell death. Apoptosis plays many other roles in your life. The dead cells that form the outermost layer of your skin and those from the uterine wall that are lost during menstruation have undergone apoptosis. White blood cells live only a few months in the circulation, then undergo apoptosis. In a form of cancer called follicular large cell lymphoma, these white blood cells do not die, but continue to divide. This cancer results from a mutation that causes the overexpression of BCL2, the gene that inhibits cell death. Plants have organ identity genes. Like animals, plants have organs, for example, leaves and roots. Many plants form flowers, and many flowers are composed of four types of organs: sepals, petals, stamens, and carpels. These floral organs occur in whorls, which are groups of each organ type stacked around a central axis. The whorls develop from meristems in the shape of domes, which develop at growing points on the stem, figure 20. How is the identity of a particular whorl different? The answer appears to lie in the activities of a group of genes. These genes have been best described in Arabidopsis thaliana. Plant is very useful for studies of development because of its small size, about 25 centimeters, abundant seed production, over 1,000 seeds per plant, rapid development, from seed to plant to seed in 6 weeks, and small genome. Finally, it is easy to produce mutations in this plant by treating the seeds with mutagens. The development of the flower begins with the meristem, which contains undifferentiated cells. Within this seemingly homogeneous cell population, individual cells sense their position and differentiate into the whorls. This happens through the expression of 3 organ identity genes, which code for proteins that act in combination with one another. Gene I is expressed in whorls 1 and 2, which form sepals and petals, respectively. Gene B is expressed in whorls 2 and 3, which form petals and stamens, respectively. Gene C is expressed in whorls 3 and 4, which form stamens and carpels, respectively. There are 2 lines of experimental evidence for this model, figure 12b. Institute for Advanced Studies, Jodhpur 11. Loss of function mutations, for example, a mutation in gene A results in no sepals or petals. Function mutations, for example, the promoter for gene C can be coupled to gene A in this case, I is expressed in all 4 whorls, resulting in only sepals and petals. Genes A, B, and C code for subunits of transcription factors, which are active as dimers. Gene regulation in these cases is combinatorial, that is, the composition of the dimer determines which other genes will be activated by the transcription factor. For example, a dimer made up only of transcription factor would activate transcription of the genes that make sepals, a dimer made up of on B would result in petals, and so forth. A common feature of the A, B, and C proteins, as well as many other plant transcription factors, is a DNA binding domain called the MADS box, named for homologous regions found in 4 genes in yeast and in 2 in plants and humans, that all encode a similar amino acid sequence. These 200 amino acid proteins also have domains for interaction with other proteins. In addition to being fascinating to biologists, plant organ identity genes have caught the eye of horticultural and agricultural scientists. Flowers filled with petals instead of stamens and carpels often have mutations of the C genes. Many of the foods that make up the human diet, such as the grains of wheat, rice, and corn, come from fruits and seeds. These fruits and seeds form from the carpels, the female reproductive organs, of the flower. Genetically modifying the number of carpels on a particular plant could increase the amount of grain a crop could produce. A gene called leafy codes for a protein that controls the transcription of the ABC genes. Plants with a mutation that causes the underexpression of leafy are just that, they make leaves, but no flowers. The protein product of this gene acts as a transcription factor stimulating genes A, B, and C so that they produce flowers, figure 13. This finding, too, has practical applications. It usually takes 6 to 20 years before a citrus tree produces flowers, and thus the fruits we eat. Scientists have made an orange tree transgenic for leafy coupled to a strongly expressed promoter, which flowers and fruits years earlier than a normal tree. Morphogen gradients provide positional information. In development, cells often need to know where they are with respect to the body as a whole, as in the case of the whorls in the flowers just described. This spatial sense is called positional information. Positional information usually comes in the form of a signal, called a morphogen, that diffuses from one group of cells down a body axis, setting up a concentration gradient. There are 2 requirements for a signal to be considered a morphogen. It must directly affect target cells, rather than triggering a secondary signal that affects target cells. Different concentrations of the signal must cause different effects. The development of the vertebrate limb provides us with an example of a morphogen in action. The limb develops from a round bud. The cells that become the bones and muscles of the limb must receive positional information. If they do not, the limbs will be totally disorganized. Imagine fingers growing out of your shoulders. The group of cells at the posterior base of the bud, just where it joins the body wall, makes a morphogen called BMP2, whose gradient determines the anterior-posterior, thumb to little finger, axis of the developing limb. Cells getting the highest dose of BMP2 make the thumb, and the smallest dose results in the little finger. The different concentrations of morphogens act through differential regulation of gene expression in their target cells. The model organism often used for studying this process has been the fruit fly. The role of differential gene expression in establishing body segmentation. Insects such as the fruit fly Drosophila melanogaster develop a highly modular body composed of different types of segments. Complex interactions of different sets of genes underlie the pattern formation of segmented bodies. Unlike body segments of segmented worms such as earthworms, which are all essentially alike, the segments of the Drosophila body are clearly different from one another. The adult fly has an anterior head composed of several fused segments, 3 different thoracic segments, and 8 abdominal segments at the posterior end. In the Drosophila larva, the thoracic and abdominal segments all appear to be similar, but they have already received their instructions to form these specialized adult segments. Several types of genes are expressed sequentially in the embryo to define these segments. The first step in this process is to establish the polarity of the embryo. Maternal effect genes encode morphogens that determine polarity. Like those of the sea urchin, Drosophila eggs and larvae are characterized by unevenly distributed cytoplasmic determinants. These molecules, which include both mRNAs and proteins, are the products of specific maternal effect genes. The maternal effect genes are transcribed in the mother's ovarian cells, which surround and nurture the developing egg and deliver the gene products to specific regions of the egg as it forms. Maternal effect genes exert their effects on the embryo regardless of the genotype of the father. Their products establish the dorsal ventral, back belly, and anterior posterior, head-tail axes of the embryo. Institute for Advanced Studies, Jodhpur 12. The fact that these morphogens specify these axes was established by the results of experiments in which cytoplasm was transferred from one egg to another. Males that are homozygous for a particular mutation of the maternal effect gene bicoid produce larvae with no head and no thorax. However, if the eggs of these females are inoculated at the anterior end with cytoplasm from the anterior region of a wild-type egg, the treated eggs develop into normal larvae. 3. Removal of 5% or more of the cytoplasm from the anterior end of a wild-type egg results in an abnormal larva that looks like a bicoid mutant larva. Another maternal effect gene, nanos, plays a comparable role in the development of the posterior end of the larva. Eggs from homozygous nanos mutant females develop into larvae with missing abdominal segments. Injecting cytoplasm from the posterior region of a wild-type egg into a nanos mutant egg allows normal development. These findings, in wild-type larvae, the overall framework of the anterior-posterior axis is laid down by the activity of these two maternal effect genes, figure 14. After the axes of the embryo are determined, the next step in pattern formation is the determination of the larval segments. Segmentation and homeotic genes act after the maternal effect genes. The number, boundaries, and polarity of the larval segments are determined by proteins encoded by the segmentation genes. These genes are expressed when there are about 6,000 nuclei in the embryo. These nuclei all look the same, but in terms of gene expression, they are not. The products of the maternal effect genes set the segmentation genes in motion. 3 classes of segmentation genes act, one after the other, to regulate finer and finer details of the segmentation pattern, figure 15. Gap genes organize broad areas along the anterior-posterior axis. Mutations in gap genes result in gaps in the body plan, the omission of several larval segments. Pair rule genes divide the embryo into units of 2 segments each. Mutations in pair rule genes result in embryos missing every other segment. Segment polarity genes determine the boundaries and anterior-posterior organization of the segments. Mutations in segment polarity genes can result in segments in which posterior structures are replaced by reversed mirror image anterior structures. Finally, after the basic pattern of segmentation has been established by the segmentation genes, differences between the segments are mediated by the activities of homeotic genes. These genes are expressed in different combinations along the length of the body and tell each segment what to become. Homeotic genes are analogous to the organ identity genes of plants. Maternal effect, segmentation, and homeotic genes interact to build a Drosophila larva step by step, beginning with the unfertilized egg. Drosophila development results from a transcriptionally controlled gene cascade. Striking and important observations about development in Drosophila and in other animals is that it results from a sequence of changes, with each change triggering the next. This sequence, or cascade, is largely controlled at the levels of transcription and translation. Most unfertilized eggs are storehouses of mRNAs, which are supplied by the mother to support protein synthesis during the early stages of embryonic development. Indeed, zygotes and early embryos do not. Institute for Advanced Studies, Jodhpur 13. Carry out transcription. Only after several cell divisions does transcription begin, forming the mRNAs needed for later development. Cytoplasmic segregation of the prefabricated mRNAs in the egg provides positional information. Before the Drosophila egg is fertilized, mRNA for the bicoid protein is localized at the end that is destined to become the anterior end of the fly. After the egg is fertilized and laid, nuclear divisions begin. In Drosophila, cytokinesis does not begin right away. Until the 13th nuclear division, the embryo is a single, multinucleated cell called a syncytium. At this early point, bicoid mRNA is translated, forming bicoid protein, which diffuses away from the anterior end, establishing a gradient. At the posterior end, the nanos protein forms a gradient in the other direction. Thus, nucleus in the developing embryo is exposed to a different concentration ratio of bicoid and nanos proteins. 2 morphogens regulate the expression of the gap genes, although in different ways. The bicoid protein affects their transcription, while the nanos protein affects their translation. The high concentrations of bicoid protein in the anterior portion of the egg turn on a gap gene called hunchback, while simultaneously turning off another gap gene, crupple. Nano of the posterior end reduces the translation of hunchback, so a difference in the concentration of these 2 gap genes products at the 2 ends is established. The proteins encoded by the gap genes control the expression of the pair rule genes. Many proteins, in turn, encode transcription factors that control the expression of the segment polarity genes, giving rise to a complex, striped pattern, see figure 15, of expression that foreshadows the segmented body plan of Drosophila. By this point, each nucleus of the embryo has been exposed to a distinct set of transcription factors. The segmented body pattern of the larva has been established even before any sign of segmentation is visible. When the segments do appear, they are not all identical, because the homeotic genes specify the different structural and functional properties of each segment. Each homeotic gene is expressed over a characteristic portion of the embryo. Let's turn now to the homeotic genes and see how their mutation can alter the course of development. Homeotic mutations produce changes in segment identity. 2 bizarre mutations in Drosophila are the antennapedia mutation, in which legs grow in place of antennae, figure 16, and the bithorax mutation, in which an extra pair of wings grows in a thoracic segment. Lewis at the California Institute of Technology found that antennapedia and bithorax were mutations not of isolated genes, but of 2 adjacent clusters of genes that determine the identity of body segments. Moreover, the genes in these clusters were lined up along the chromosome in the same order as the segments they determined. From left to right, genes in the first cluster specified anterior body segments, starting with genes for the different head segments and ending with thoracic segments. The second cluster began with a gene specifying the last thoracic segment, followed by a gene for the anterior abdominal segments, and ended with a gene for the posterior abdominal segments. Lewis hypothesized that all of these genes might have come from the duplication of a single gene in an ancestral, unsegmented organism. Molecular biologists confirmed Lewis' hypothesis using nucleic acid hybridization. Several scientists found that a probe for a sequence in one of the genes of one cluster bound not only to its own gene, but also to adjacent genes in its cluster and to genes in the other homeotic cluster. In other words, this DNA sequence is common to all the homeotic genes in both clusters. Genes encode transcription factors. 180 base pair DNA sequence that is common to the bithorax and Antennopedidae gene clusters is called the homeobox. It encodes a 60 amino acid sequence, called the homeodomain, that binds to DNA. The homeodomain turns out to be present in other proteins involved in Drosophila pattern formation, such as bicoid. In all cases, the homeodomain portion of the protein has a helix-turn helix motif, see figure 15. Each type of homeodomain recognizes a specific DNA sequence in the promoter of its target genes. The bicoid homeodomain, for example, recognizes TCC-TAA-TCCC. What do homeodomain proteins do when they recognize their target sequence in DNA? Not surprisingly, they are transcription factors. The bicoid protein, for example, binds to promoters of the GAP gene hunchback, activating its transcription. The hunchback protein is also a transcription factor, which binds to enhancers of genes involved in head and thorax formation. In this way, the homeodomain proteins produce a cascade of events that controls Drosophila development. Homeobox genes are found in many animals, including humans. They play a role in development similar to the role the MADS box genes play in plants. Institute for Advanced Studies, Jodhpur 14. B. Gametogenesis, fertilization and early development. Sexual reproduction. A large time and energy budgets of sexually reproducing animals goes into mating, which exposes them to predation, can result in physical damage, and detracts from other useful activities, such as feeding and caring for existing offspring. Furthermore, mating requires that resources be used to maintain a large population of males that do not bear offspring. In spite of all of these disadvantages, there is an overwhelming evolutionary advantage to sexual reproduction. It produces genetic diversity. Sexual reproduction requires the joining of two haploid sex cells to form a diploid individual. These haploid cells, or gametes, are produced through gametogenesis, a process that involves meiotic cell divisions. Two events in meiosis contribute to genetic diversity, crossing over between homologous chromosomes and the independent assortment of chromosomes. Sexual reproduction itself also contributes to genetic diversity. Gain among the gametes of a single individual and the genetic variation between any two parents produce an enormous potential for genetic variation between any two offspring of a sexually reproducing pair of individuals. This genetic diversity is the raw material for natural selection. Sexual reproduction in animals consists of three fundamental steps. Gametogenesis, making gametes. Mating, adding gametes together. Fertilization, getting gametes to fuse. There is not a great deal of diversity in gametogenesis when we compare different groups of animals. Processes of fertilization are also rather similar in widely different species. Therefore, the discussion of gametogenesis that follows focuses primarily on mammals, and our discussion of fertilization mainly deals with sea urchins. The facts would not be dramatically different if we focused on a different group of animals. Adaptations for mating, on the other hand, show incredible anatomical, physiological, and behavioral diversity. Eggs and sperm formed through gametogenesis. Gametogenesis occurs in the gonads, which are testes singular, testis in males and ovaries in females. The tiny gametes of males, called sperm, move by beating the flagella. The larger gametes of females, called eggs or ova singular, ovum or non-motile, see figure 1. Institute for Advanced Studies, Jodhpur 15. Gametes are produced from germ cells, which have their origin in the earliest cell divisions of the embryo and remain distinct from the rest of the body. All other cells of the embryo are called somatic cells. Germ cells are sequestered in the body of the embryo until its gonads begin to form. Stem cells then migrate to the gonads, where they take up residence and proliferate by mitosis, producing ogoniate singular, ogonium in females and spermatogoniate singular, spermatogonium in males. Ogonia and spermatogonia, which are diploid, also multiply by mitosis, eventually producing primary oocytes and primary spermatocytes, which are still diploid cells. Meiosis, the next step in gametogenesis, reduces the chromosomes to the haploid number, and the resulting haploid cells eventually mature into sperm and ova. Although the steps of meiosis are very similar in males and females, there are some significant differences in gametogenesis between the sexes. Spermatogenesis produces sperm. Primary spermatocytes undergo the first meiotic division to form secondary spermatocytes, which are haploid. The second meiotic division produces 4 haploid spermatids for each primary spermatocyte that entered meiosis, figure 1a. In mammals, these cells remain connected by cross bridges of cytoplasm after each division. The reason that mammalian spermatocytes remain in cytoplasmic contact throughout their development probably is the asymmetry of sex chromosomes in males. Half the secondary spermatocytes receive an X chromosome, the other half a Y chromosome. The Y chromosome contains fewer genes than the X chromosome, and apparently some of the products of genes found only on the X chromosome are essential for spermatocyte development. Remaining in cytoplasmic contact, all 4 spermatocytes can share the gene products of the X chromosomes, even though only half of them have an X chromosome. A spermatid bears little resemblance to a mature sperm. Through further differentiation, however, it will become compact, streamlined, and motile. Oogenesis produces eggs. Oogonia, like spermatogonia, proliferate through mitosis. The resulting primary oocytes immediately enter prophase of the first meiotic division. In many species, including humans, the development of the oocyte is arrested at this point and may remain so for days, months, or years. In contrast, there is no arrest during male gametogenesis, which goes steadily to completion once the primary spermatocyte has differentiated. In the human female, as we will see, some primary oocytes may remain in arrested prophase for up to 50 years. During this prolonged prophase, or shortly before it ends, the primary oocyte undergoes its major growth phase. It grows larger due to increased production of ribosomes, RNA, cytoplasmic organelles, and energy stores. At this, the primary oocyte acquires all the energy, raw materials, and RNA that the egg will need to survive its first cell divisions after fertilization. In fact, the nutrients in the egg will have to maintain the embryo until it is either nourished by the maternal system or can feed on its own. When a primary oocyte resumes meiosis, its nucleus completes the first meiotic division near the surface of the cell. The daughter cells of this division receive grossly unequal shares of cytoplasm. This asymmetry represents another major difference from spermatogenesis, in which cytoplasm is apportioned equally. The daughter cell that receives almost all the cytoplasm becomes the secondary oocyte, and the one that receives almost none forms the first polar body, figure 1b. Second meiotic division of the large secondary oocyte is also accompanied by an asymmetrical division of the cytoplasm. One daughter cell forms the large, haploid ootid, which eventually differentiates into a mature ovum, and the other forms the second polar body. Polar bodies degenerate, so the end result of oogenesis is only one mature egg for each primary oocyte that entered meiosis. However, that egg is a very large, well-provisioned cell. A second period of arrested development occurs after the first meiotic division forms the secondary oocyte. The egg may be expelled from the ovary in this condition. In many species, including humans, the second meiotic division is not completed until the egg is fertilized by a sperm. Fertilization is the union of sperm and egg. The union of the haploid sperm and the haploid egg creates a single diploid cell, called a zygote, which will develop into an embryo. Fertilization does more, however, than just restore the full genetic complement of the animal. The events and processes associated with fertilization help eggs and sperm get together, prevent the union of sperm and eggs of different species, guarantee that only one sperm will enter an egg, and activate the egg metabolically. Fertilization involves a complex series of events. The sperm and the egg recognize each other. The sperm is activated so that it is capable of gaining access to the plasma membrane of the egg. The plasma membranes of the sperm and the egg fuse. The egg blocks entry by additional sperm. The egg is metabolically activated and stimulated to start development. The egg and sperm nuclei fuse to create the diploid nucleus of the zygote. Specificity in sperm-egg interactions. Specific recognition molecules mediate interactions between sperm and eggs. Molecules ensure that the activities of sperm are directed toward eggs and not other cells. The latter function is particularly important in aquatic species that release eggs and sperm into the surrounding water. The sea urchin is such a species, and its mechanisms of fertilization have been well studied. The eggs of sea urchins and various other marine invertebrates release chemical attractants that increase The eggs of sea urchins and various other marine invertebrates release chemical attractants that increase. Institute for Advanced Studies, Jodhpur 16. The motility of sperm and cause them to swim toward the egg. These chemical attractants are species-specific. For example, eggs of one species of sea urchin release a peptide consisting of 14 amino acids. As this peptide diffuses from the egg, it binds to receptors on the sperm of the same species. The sperm respond by increasing their mitochondrial respiration and their motility. Before exposed to the peptide, the sperm swim in tight little circles, but after binding the peptide, they swim energetically up the concentration gradient of the peptide until they reach the egg that is releasing it. The peptide released by eggs of one species of sea urchin does not bind to receptors on sperm of other species. When sperm reach an egg, they must get through two protective layers before they can fuse with the egg plasma membrane. The eggs of sea urchins are covered with a jelly coat, which surrounds a proteinaceous vitelline envelope. The sperm's assault on these protective layers depends on a membrane-enclosed structure called an acrosome containing enzymes and other proteins. The acrosome is located at the front of the sperm head, where it forms a cap over the nucleus, figure 2. When in contact with an egg of its own species, substances in the jelly coat trigger an acrosomal reaction, which begins with the breakdown of the plasma membrane covering the sperm head and the underlying acrosomal membrane. The acrosomal enzymes are released, and they digest a hole through the jelly coat. Next, a structure called an acrosomal process extends out of the head of the sperm. The acrosomal process forms from globular actin proteins behind the acrosome, which polymerize when the acrosomal membrane breaks down. The acrosomal process extends through the remainder of the jelly coat to make contact with the vitelline envelope. The acrosomal process is coated with a membrane-bound protein called bindin. Different species have different kinds of bindin molecules. The plasma membrane of the egg has species-specific bindin receptors that extend through the vitelline envelope. The of acrosomal bindin with these receptors stimulates the egg plasma membrane to form a fertilization cone that engulfs the sperm head, bringing it into the egg cytoplasm. In animals that practice internal fertilization, mating behaviors help guarantee species specificity, but egg-sperm recognition mechanisms still exist. The mammalian egg, for example, is surrounded by a thick layer called the cumulus, which consists of a loose assemblage of maternal cells in a gelatinous matrix, figure 2. Beneath the cumulus is a glycoprotein envelope called the zona pellucida, which is functionally similar to the vitelline envelope of sea urchin eggs. When mammalian sperm are deposited in the female reproductive tract, they become metabolically activated and are made capable of an acrosomal reaction if they should meet an egg. An activated sperm can penetrate the cumulus and interact with the zona pellucida. Unlike the jelly coat of sea urchin eggs, the cumulus of mammalian eggs does not trigger the acrosomal reaction. When in contact with the zona pellucida, a species-specific glycoprotein in the zona binds to recognition molecules on the head of the sperm. This binding triggers the acrosomal reaction, releasing acrosomal enzymes that digest a path through the zona. When sperm head reaches the egg plasma membrane, other proteins cause the adhesion of sperm to egg plasma membrane and facilitate fusion of sperm and egg. The importance of the zona pellucida and its sperm binding molecules as a species-specific recognition mechanism was revealed in experiments on mammalian eggs and sperm in culture dishes. When the zona was stripped from human eggs and they were exposed to hamster sperm, fertilization took place, resulting in a hamster-human hybrid zygote. The zygote did not survive its first cell division because of chromosomal incompatibilities, but the experiment demonstrated that the recognition mechanism in mammalian species resides in the zona. Blocks to polyspermy in egg. Activation. The fusion of the sperm and egg plasma membranes and the entry of the sperm into the egg initiate a programmed sequence of events. The first responses to sperm entry are blocks to polyspermy, that is, mechanisms that prevent more than one sperm from entering the egg. If more than one sperm enters the egg, the resulting embryo is unlikely to survive. Blocks to polyspermy have been studied extensively in sea urchin eggs, which can be fertilized in a dish of seawater. Within seconds after a sperm enters a sea urchin egg, there is an influx of sodium ions, which changes the electric charge difference across the egg's plasma membrane. This block to polyspermy prevents the fusion of other sperm with the egg plasma membrane. The slow block to polyspermy takes about a minute and results from the release of calcium, figure 3. Before fertilization, the vitelline envelope is bonded to the egg plasma membrane. Just under the plasma membrane are vesicles called cortical granules, which contain enzymes and other proteins. The sea urchin egg, like all animal cells, contains calcium ions that are sequestered in the endoplasmic reticulum. Sperm binding to the sea urchin egg stimulates the release of calcium from the egg's endoplasmic reticulum. The increase in cytoplasmic. Institute for Advanced Studies, Jodhpur 17. Calcium causes the egg's cortical granules to fuse with the plasma membrane and release their contents. Cortical granule enzymes break the bonds between the vitelline envelope and the plasma membrane, and other proteins released from the cortical granules attract water into the space between them. As a result, the vitelline envelope rises to form a fertilization envelope. Cortical granule enzymes also degrade sperm binding molecules on the surface of the fertilization envelope and cause it to harden. The fertilization envelope prevents additional sperm from contacting the egg. The slow block to polyspermy in the sea urchin is mediated by the phosphatidylinositol bisphosphate (PIP2) second messenger system. Action of the bindin receptors activates phospholipase C, which cleaves PIP2 in the egg plasma membrane, releasing inositol triphosphate IP3 into the egg cell cytoplasm. IP3 diffuses to the endoplasmic reticulum, where it opens calcium channels. In males, sperm entry does not seem to cause a rapid change in membrane potential, but it triggers the PIP2 second messenger system, resulting in several events. Calcium is released from the endoplasmic reticulum, and as in the sea urchin, the increased calcium causes the cortical granules to fuse with the egg plasma membrane. A physation envelope does not form around the mammalian egg, but the cortical granule enzymes destroy the molecules in the zona pellucida that bind sperm. The rise in cytoplasmic calcium also activates the egg's metabolism and signals it to complete meiosis. The pH of the egg's cytoplasm increases, its oxygen consumption rises, protein synthesis increases, and DNA synthesis is initiated. The stage is set for the first cell division. Anatomical and behavioral adaptations bring eggs and sperm together. Just seen, sexual reproduction requires the production of haploid gametes, gametogenesis, and the joining together of those gametes to form a diploid zygote, fertilization. Mating, the step in between these two processes, gets eggs and sperm close enough together so that fertilization can occur. The simplest distinction in mating systems is whether fertilization occurs externally or internally. External fertilization. In an aquatic environment, animals can bring their gametes together by simply releasing them into the water. This practice, called external fertilization, is common among simple aquatic animals that are not very mobile. Such animals may produce huge numbers of gametes. A female oyster, for example, may produce 100 million eggs in a year, and the number of sperm produced by a male oyster is astronomical. But numbers alone do not guarantee that gametes will meet. The reproductive activities of the males and females of a population must be synchronized, since released gametes have a limited lifespan. Seasonal breeders may use day length, changes in temperature, or changes in weather to time their production and release of gametes. Social stimulation is also important. Sexual activity by one member of a population can stimulate others to engage in it. Behavior can play an important role in bringing gametes together even when fertilization is external. Many species travel great distances to congregate with potential mates and release their gametes at the same time in a suitable environment. Salmon are an extreme example, traveling hundreds of miles to spawn in a stream where they hatched. Internal fertilization. Terrestrial animals cannot simply release their gametes into the environment. Sperm can move only through liquid, and delicate gametes released into air would dry out and die. Terrestrial animals avoid these problems by releasing sperm directly into the female reproductive tract. This practice is called internal fertilization. Animals have evolved an incredible diversity of behavioral and anatomical adaptations for internal fertilization. As we saw above, gametogenesis occurs in the gonads, which are the primary sex organs. All of the additional anatomical components of an animal's reproductive system are called accessory sex organs. An obvious accessory sex organ in the males of many species is the penis, which enables the male to deposit sperm in the female's reproductive tract. Accessory sex organs include a variety of glands, tubules, ducts, and other structures. Copulation is the physical joining of male and female accessory sex organs. Transfer of sperm and internal fertilization can also be indirect. Males of many invertebrate species, e.g., mites and scorpions, and a few vertebrates, e.g., salamanders, deposit spermatophores, packets of sperm, in the environment. When a female mite encounters a spermatophore from a potential mate, she straddles it and opens a pair of plates in her abdomen. Institute for Advanced Studies, Jodhpur 18. So that the tip of the spermatophore enters her reproductive tract and allows the sperm to enter. Male squids and spiders play a more active role in spermatophore transfer. The spider secretes a drop containing sperm onto a bit of web, then uses a special structure on his foreleg to pick up the sperm containing web and insert it through the female's genital opening. Male squids use one specialized tentacle to pick up a spermatophore and insert it into the female's genital opening. Most male insects copulate and transfer sperm to the female's vagina through a penis. The genitalia, external sex organs, of insects often have species-specific shapes that match in a lock and key fashion. This mechanism ensures a tight, secure fit between the mating pair during the prolonged period of sperm transfer. In some sex species in which females mate with more than one male, the males have elaborate structures on their penises that can scoop sperm deposited by other males out of a female's reproductive tract, replacing it with their own. A single body can function as both male and female. In most species, gametes are produced by individuals that are either male or female. Species that have separate male and female members are called dioecious species, from the Greek for two houses. In some species, however, a single individual may produce both sperm and eggs. Such species are called monoecious, one house, or hermaphroditic species. Almost all invertebrate groups contain some hermaphroditic species. An earthworm is an example of a simultaneous hermaphrodite, meaning that it is both male and female at the same time. When two earthworms mate, they exchange sperm, and as a result, the eggs of each are fertilized. Some vertebrates, such as the anemone fish, are sequential hermaphrodites, meaning that an individual may function as a male or as a female at different times in its life. What is the selective advantage of hermaphroditism? Some simultaneous hermaphrodites, such as parasitic tapeworms, have a low probability of meeting a potential mate. Even though a tapeworm may be large and cause lots of trouble for its host, it may be the only tapeworm in the host. Tapeworms can fertilize their own eggs. Most simultaneous hermaphrodites must mate with another individual, but since each member of the population is both male and female, the probability of encountering a possible mate is double what it would be in monoecious species. In some sequential hermaphrodites, all siblings are either male or female at the same time, thus reducing the incidence of inbreeding. The evolution of vertebrate reproductive systems parallels the move to land. The earliest vertebrates evolved in aquatic environments. The closest living relatives of those earliest vertebrates are modern-day fishes. They remain exclusively aquatic animals, and most practice external fertilization. The most primitive of the fishes, the lampreys and hagfishes, simply release their gametes into the environment. In most fishes, however, mating behaviors bring females and males into close proximity at the time of gamete release. In rays, fins have evolved into claspers that hold the male and female together and enable sperm to be transferred directly into the female reproductive tract. Amphibians were the first vertebrates to live in terrestrial environments. They dealt with the challenge of a dry environment by returning to water to reproduce, as most amphibians still do today. Reptiles were the first vertebrate group to solve the problem of reproduction in the terrestrial environment. Their solution, the amniote egg, is shared with the birds. A good example is the chicken egg, which contains a supply of food, yolk, and water for the developing embryo. A hard shell protects the embryo and impedes water loss while allowing the diffusion of oxygen and carbon dioxide. The eggshell creates an obvious problem for fertilization. However, sperm cannot penetrate the shell, so they have to reach the egg before the shell forms. Hence, internal fertilization and the evolution of accessory sex organs were necessary for the evolution of the amniote egg. Fertilization and the evolution of accessory sex organs were necessary for the evolution of the amniote egg. Male snakes and lizards have paired hemipenes, which can be filled with blood and thereby extruded from the male's body. Only one hemipene is inserted into the female's reproductive tract at a time. It is usually rough or spiny at the end to achieve a secure hold while sperm are transferred down a groove on its surface. Retractor muscles pull the hemipene back into the male's body when mating is completed. Some birds, mostly more primitive species, have erectile penises that channel sperm along a groove into the female's reproductive tract. Species with more recent evolutionary origins, however, do not have erectile penises. Instead, the male and female simply bring their genital openings close together to transfer sperm. Usually this involves the male standing on the female's back. All mammals practice internal fertilization, but except for the monotremes, they have done away with the shelled egg. They retain the developing embryo in the female reproductive tract, at least through the early stages of development. Mammalian species vary enormously as to the developmental stage of their offspring at the time of birth. Reproductive systems are distinguished by where the embryo develops. Two patterns of care and nurture of the embryo have evolved in animals: oviparity, egg-laying, and viviparity, live-bearing. Oviparous animals lay eggs in the environment, and their embryos develop outside the mother's body. Oviparity is possible because eggs are stocked with abundant nutrients to supply the needs of the embryo. Thus terrestrial animals, such as insects, reptiles, and birds, protect their eggs from desiccation with tough, waterproof membranes or shells. Some oviparous animals engage in various forms of parental behavior to protect their eggs, but until the eggs hatch, the embryos depend entirely on the nutrients stored in the egg. The only oviparous mammalian species are the monotremes, the echidnas and the duck-billed platypus. Viviparous animals retain the embryo within the mother's body during its early developmental stages. All mammals, except monotremes, are viviparous. There are examples of viviparity in all other vertebrate groups except the crocodiles, turtles, and birds. Even some sharks retain fertilized eggs in their bodies and give birth to free-living offspring. But there is a big difference between viviparity in mammals and in other species. Mammals have this portion of the female reproductive tract, called the uterus or womb, that holds the embryo and interacts with it to produce a placenta, which enables the exchange of nutrients and wastes between the blood of the mother and that of the embryo. Non-mammalian viviparous animals simply retain fertilized eggs in the mother's body until they hatch. These embryos still receive nutrition from stores in the egg, so this reproductive adaptation is called ovoviviparity. Development begins with fertilization. Fertilization is the union of a haploid sperm and a haploid egg to produce a diploid zygote. Fertilization does more, however, than just restore a full complement of maternal and paternal genes. The entry of a sperm into an egg activates the egg metabolically and initiates a rapid series of cell divisions that produce a multicellular embryo. Also, in many species, the point of entry of the sperm creates an asymmetry in the radially symmetrical egg. This asymmetry is the initiating event that enables a bilateral body plan to emerge from the radial symmetry of the egg. We take a closer look at the cellular and molecular interactions of sperm and egg that result in the first steps of development. The sperm and the egg make different contributions to the zygote. Nearly all of the cytoplasm of the zygote comes from the egg. Egg cytoplasm is well stocked with nutrients, ribosomes, and a variety of molecules, including mRNAs. If the sperm's mitochondria degenerate, all of the mitochondria, and therefore all of the mitochondrial DNA, in the zygote come from the mother. In addition to its haploid nucleus, the sperm makes one other important contribution to the zygote in some species, a centriole. This centriole becomes the centrosome of the zygote, which produces the meiotic spindles for subsequent cell divisions. It had long been assumed that the one thing that sperm and egg contributed equally to the zygote was their haploid nuclei. However, we now know that even though they are equivalent in terms of genetic material, mammalian sperm and eggs are not equivalent in terms of their roles in development. In the laboratory, it is possible to construct zygotes in which both haploid nuclei come from the mother or both come from the father. In neither case does development progress normally. Apparently, in mammals at least, certain genes involved in development are active only if they come from a sperm, and others are active only if they come from an egg. This phenomenon, called genomic imprinting. Fertilization causes rearrangements of egg cytoplasm. The entry of the sperm into the egg stimulates changes in and rearrangements of the egg cytoplasm that establish the polarity of the embryo. The nutrients and molecules in the cytoplasm of the zygote are not homogeneously distributed, and therefore, they are not divided equally among all daughter cells when cell divisions begin. This distribution of cytoplasmic factors sets the stage for the signal transduction cascades that orchestrate the sequential steps of development, determination, differentiation, and morphogenesis. Let's examine these earliest developmental events in the frog, an organism in which they have been well studied. The rearrangements of egg cytoplasm in some frog species are easily observed because of pigments in the egg cytoplasm. The nutrient molecules in an unfertilized frog egg are dense, and they are therefore concentrated by gravity in the lower half of the egg, which is called the vegetal hemisphere. The haploid nucleus of the egg is located at the opposite end of the egg, in the animal hemisphere. The postcortical cytoplasm of the animal hemisphere is heavily pigmented, and the underlying cytoplasm has more diffuse pigmenting. The vegetal hemisphere is not pigmented. The surface of the frog egg has specific sperm binding sites located only in the animal hemisphere, so sperm always enter the egg in that hemisphere. When a sperm enters, the cortical cytoplasm rotates toward the site of sperm entry. This rotation reveals a band of diffusely pigmented cytoplasm on the side of the egg opposite the site of sperm entry. This band, called the gray crescent, will be the site of important developmental events. Figure 4. The cytoplasmic rearrangement The vegetal hemisphere is not pigmented. The surface of the frog egg has specific sperm binding sites located only in the animal hemisphere, so sperm always enter the egg in that hemisphere. The vegetal hemisphere is not pigmented. The surface of the frog egg has specific sperm binding sites located only in the animal hemisphere, so sperm always enter the egg in that hemisphere. When a sperm enters, the cortical cytoplasm rotates toward the site of sperm entry. This rotation reveals a band of diffusely pigmented cytoplasm on the side of the egg opposite the site of sperm entry. This band, called the gray crescent, will be the site of important developmental events. Figure 4. The cytoplasmic rearrangements that create the gray crescent bring different regions of cytoplasm into contact on opposite sides of the egg. Therefore, bilateral symmetry is imposed on what was a radially symmetrical egg. In the up-down difference of the animal and vegetal hemispheres, the movement of the cytoplasm sets the stage for the creation of the anterior-posterior and left-right axes. In the frog, the site of sperm entry will become the ventral belly region of the embryo, and the gray crescent will become the dorsal back region. Since the gray crescent also marks the posterior end of the embryo, these relationships specify the anterior-posterior and left-right axes as well. Institute for Advanced Studies, Jodhpur 20. Rearrangements of egg cytoplasm set the stage for determination. The molecular mechanisms underlying the first steps in frog embryo formation are beginning to be understood. Sperm centriole rearranges the microtubules in the vegetal hemisphere cytoplasm into a parallel array that presumably guides the movement of the cortical cytoplasm. Organelles and certain proteins from the vegetal hemisphere move to the gray crescent region even faster than the cortical cytoplasm rotates. As a result of these movements of cytoplasm, proteins, and organelles, changes in the distribution of critical developmental signals occur. A key transcription factor in early development is beta-catenin, which is produced from maternal mRNA and is found throughout the cytoplasm of the egg. Also present throughout the egg cytoplasm is a protein kinase called GSK3, which phosphorylates and thereby targets beta-catenin for degradation. However, an inhibitor of GSK3 is segregated in the vegetal cortex of the egg. After sperm entry, this inhibitor is moved along microtubules to the gray crescent, where it prevents the degradation of beta-catenin. As a result, the concentration of beta-catenin is higher on the dorsal side than on the ventral side of the developing embryo. Figure 5. It supports the hypothesis that beta-catenin is a key player in the cell-cell signaling cascade that begins the process of cell determination and the formation of the embryo in the region of the gray crescent. Can be cell-cell signaling, there must be multiple cells, so let's turn first to the early series of cell divisions that transforms the zygote into a multicellular embryo. Cleavage, repackaging the cytoplasm. The transformation of the diploid zygote into a mass of cells occurs through a rapid series of cell divisions, called cleavage. Because the cytoplasm of a zygote is not homogeneous, these first cell divisions result in the differential distribution of nutrients and cytoplasmic determinants among the cells of the early embryo. In most animals, cleavage proceeds with rapid DNA replication and mitosis, but no cell growth and little gene expression. The embryo becomes a solid ball of smaller and smaller cells, called a morolla from the Latin word for mulberry. Eventually, this ball forms a central fluid-filled cavity called a blastocele, at which point the embryo is called a blastula. Its individual cells are called blastomeres. Pattern of cleavage, and therefore the form of the blastula, is influenced by two major factors. First, the amount of nutrient material, or yolk, stored in the egg differs among species. Second is the pattern of cell divisions by impeding the pinching in of the plasma membrane to form a cleavage furrow between the ductaire cells. Second, cytoplasmic determinants stored in the egg by the mother guide the formation of meiotic spindles and the timing of cell divisions. The amount of yolk influences cleavage. In egg with little or no yolk, there is little interference with cleavage furrow formation, and all the daughter cells are of similar size. The sea urchin egg provides an example. Figure 6a. More yolk means more resistance to cleavage furrow formation. Therefore, cell divisions progress more rapidly in the animal hemisphere than in the vegetal hemisphere, where the yolk is concentrated. As a result, the cells derived from the vegetal hemisphere are fewer and larger. The frog egg provides an example of this pattern. Figure 6b. In spite of this difference between sea urchin and frog eggs, the cleavage furrows completely divide the egg mass in both cases. Thus, these animals are said to have complete cleavage. In contrast, in eggs that contain a lot of yolk, such as the chicken egg, the cleavage furrows do not penetrate the yolk. As a result, cleavage is incomplete, and the embryo forms as a disc of cells, called a blastodisc, on top of the yolk mass. Figure 6c. This type of incomplete cleavage, called discoidal cleavage, is Institute for Advanced Studies, Jodhpur 21. Common in fishes, reptiles, and birds. Another type of incomplete cleavage, called superficial cleavage, occurs in insects such as the fruit fly Drosophila. In the insect egg, the mass of yolk is centrally located. Figure 6d. Early in development, cycles of mitosis occur without cytokinesis. The resulting nuclei migrate to the periphery of the egg, and after several more meiotic cycles, the plasma membrane of the egg grows inward, partitioning the nuclei into individual cells. The orientation of meiotic spindles influences the pattern of cleavage. Positions of the meiotic spindles during cleavage are not random, rather, they are defined by cytoplasmic determinants that were produced from the maternal genome and stored in the egg. The orientation of the meiotic spindles determines the planes of cleavage and, therefore, the arrangement of the daughter cells. If meiotic spindles of successive cell divisions form parallel or perpendicular to the animal-vegetal axis of the zygote, the cleavage pattern is radial, as in the sea urchin and the frog. In these organisms, the first two cell divisions are parallel to the animal-vegetal axis and the third is perpendicular to it. Figure 6a, b. Another cleavage pattern, spiral cleavage, results when the meiotic spindles are at oblique angles to the animal-vegetal axis. Mollusks have spiral cleavage, and a visible expression of this is the coiling of snail shells. Cleavage in mammals is unique. Several features of mammalian cleavage are very different from those seen in other animal groups. First, the pattern of cleavage in mammals is rotational. The first cell division is parallel to the animal-vegetal axis, yielding two blastomeres. The second cell division occurs at right angles. One blastomere divides parallel to the animal-vegetal axis, while the other divides perpendicular to it. Figure 7a. Cleavage in mammals is very slow. Cell divisions are 12 to 24 hours apart, compared with tens of minutes to a few hours in non-mammalian species. Also, the cell divisions of mammalian blastomeres are not in synchrony with each other. Because the blastomeres do not undergo mitosis at the same time, the number of cells in the embryo does not progress in the regular 2, 4, 8, 16, 32, etc. progression typical of other species. Another unique feature of the slow mammalian cleavage is that the products of genes expressed at this time play roles in cleavage. Species such as sea urchins and frogs, gene expression does not occur in the blastomeres, and cleavage is directed exclusively by molecules that were present in the egg prior to fertilization. As in other animals that have complete cleavage, the early cell divisions in a mammalian zygote produce a loosely associated ball of cells. However, at about the 8-cell stage, the behavior of the mammalian blastomeres. Institute for Advanced Studies, Jodhpur 22. Changes. They change shape to maximize their surface contact with one another, form tight junctions, and become a very compact mass of cells, figure 7b. At the transition from the 16-cell to the 32-cell stage, the cells separate into two groups. The inner cell mass will become the embryo, while the surrounding cells become an encompassing sac called the trophoblast, which will become part of the placenta. Trophoblasts secrete fluid, creating a cavity blastocele with the inner cell mass at one end, see figure 7b. At this stage, the mammalian embryo is called a blastocyst to distinguish it from the blastulas of other animals. Fertilization in mammals occurs in the upper reaches of the mother's oviduct, and cleavage occurs as the zygote travels down the oviduct to the uterus. When the blastocyst arrives in the uterus, the trophoblast adheres to the endometrium, the uterine wall. This event begins the process of implantation that embeds the embryo in the wall of the uterus. In humans, implantation begins on about the 6th day after fertilization. As the blastocyst moves down the oviduct to the uterus, it must not embed itself in the oviduct wall, or the result will be an ectopic or tubal pregnancy, a very dangerous condition. Implantation is normally prevented by an external proteinaceous layer called the zona pellucida, which surrounds the egg and remains around the cleaving ball of cells. At about the time the blastocyst reaches the uterus, it hatches from the zona pellucida, and implantation can occur. Specific blastomeres generate specific tissues and organs. In all animal species, cleavage results in a repackaging of the egg cytoplasm into a large number of small cells surrounding a central cavity. Little cell differentiation occurs during cleavage, and in most non-mammalian species, none of the Little cell differentiation occurs during cleavage, and in most non-mammalian species, none of the genome of the embryo is expressed. Nevertheless, cells in different regions of the resulting blastula possess different complements of the nutrients and cytoplasmic determinants that were present in the egg. The blastocele prevents cells from different regions of the blastula from interacting, but that will soon change. Next stage of development, the cells of the blastula will move around and come into new associations with one another, communicate instructions to one another, and begin to differentiate. In animals, these movements of the blastomeres are so regular and well orchestrated that it is possible to label a specific blastomere with a dye and identify the tissues and organs that form from its progeny. Such labeling experiments produce fate maps of the blastula, figure 8. Institute for Advanced Studies, Jodhpur 23. Blastomeres become determined, committed to specific fates, at different times in different species. In some species, such as roundworms and clams, blastomeres are determined by the 8-cell stage. If one of these blastomeres is experimentally removed, a particular portion of the embryo will not form. This type of development has been called mosaic development because each blastomere seems to contribute a specific set of tiles to the final mosaic, that is the adult animal. In contrast, other species, such as sea urchins and vertebrates, have regulated development. The loss of some cells during cleavage does not affect the developing embryo because the remaining cells compensate for the loss. If some blastomeres can change their fate to compensate for the loss of other cells during cleavage and blastula formation, are those cells capable of forming an entire embryo? To a certain extent, they are. During cleavage or early blastula formation in mammals, for example, if the blastomeres are physically separated into two groups, both groups can produce complete embryos, figure 9. Since the two embryos come from the same zygote, they will be monozygotic twins, genetically identical. Non-identical twins occur when two separate eggs are fertilized by two separate sperm. Thus, while identical twins are always of the same sex, non-identical twins have a 50% chance of being the same sex. Gastrulation, producing the body plan. The blastula is typically a fluid-filled ball of cells. How does this simple ball of cells become an embryo, made up of multiple tissue layers, with head and tail? How does this simple ball of cells become an embryo, made up of multiple tissue layers, with head and tail ends and dorsal and ventral sides? Gastrulation is the process whereby the blastula is transformed by massive movements of cells into an embryo with multiple tissue layers and visible body axes. The resulting spatial relationships between tissues make possible the inductive interactions that trigger differentiation and organ formation. During gastrulation, the animal body forms 3 germ layers, also called cell layers or tissue layers. Some blastomeres move together as a sheath to the inside of the embryo, creating an inner germ layer called the endoderm. The endoderm will give rise to the lining of the digestive tract, respiratory tract, and circulatory system and make up other internal tissues such as the pancreas and liver. The cells remaining on the outside of the embryo become the outer germ layer, the ectoderm. The ectoderm will give rise to the nervous system, the skin, hair, and nails, sweat glands, oil glands, and milk secretory ducts. Other cells migrate between the endoderm and the ectoderm to become the middle germ layer, or mesoderm. The mesoderm will contribute tissues to many organs, including blood vessels, muscle, bones, liver, and heart. Such challenging and interesting questions in animal development have concerned what directs the cell movements of gastrulation and what is responsible for the resulting patterns of cell differentiation and organ formation. In the past 25 years, scientists have answered many of these questions at the molecular level. As it follows, we'll consider the similarities and differences among gastrulation in sea urchins, frogs, reptiles, birds, and mammals. Pagination of the vegetal pole characterizes gastrulation in the sea urchin. The sea urchin blastula is a simple, hollow ball of cells that is only 1 cell thick. The end of the blastula stage is marked by a dramatic slowing of the rate of mitosis, and the beginning of gastrulation is marked by a flattening of the vegetal hemisphere, figure 10. Some cells at the vegetal pole bulge into the blastocele, break free, and migrate into the cavity. These cells become primary mesenchyme cells, cells of the middle germ layer, the mesoderm. Mesenchyme cells are unconnected to one another and act as independent units, in contrast to epithelial cells, which are tightly packed into sheaths or tubes. The flattening of the vegetal pole results from changes in the shape of the individual blastomeres. These cells shift from being rather cuboidal to become wedge-shaped, with constricted outer edges and expanded inner edges. As a result of these shape changes, the vegetal pole bulges inward, or invaginates, as if someone were poking a finger into a hollow ball. Institute for Advanced Studies, Jodhpur 24. The cells that invaginate become the endoderm and form the primitive gut, the archenteron. At the tip of the archenteron more cells break free, entering the blastocele to form more mesoderm, the secondary mesenchyme. At the tip of the archenteron more cells break free, entering the blastocele to form more mesoderm, the secondary mesenchyme. The early invagination of the archenteron is due to the changes in cell shapes, but eventually it is pulled by the secondary mesenchyme cells. These cells, attached to the tip of the archenteron, send out extensions that adhere to the overlying ectoderm and contract. Where the archenteron eventually makes contact with the ectoderm, the mouth of the animal will form. The opening created by the invagination of the vegetal pole is called the blastopore. It will become the anus of the animal. What mechanisms control the various cell movements of sea urchin gastrulation? The immediate answer is that specific properties of particular blastomeres change. For example, some vegetal cells migrate into the blastocele to form the primary mesenchyme because they lose their attachments to neighboring cells. Once they bulge into the blastocele, they move by extending long processes called phylopodia along an extracellular matrix of proteins that is laid down by the ectodermal cells lining the blastocele. Deeper understanding of gastrulation requires that we discover the molecular mechanisms whereby certain blastomeres develop properties different from those of others. Cleavage divides up the cytoplasm of the egg in a very systematic way. The sea urchin blastula at the 64-cell stage is radially symmetrical, but it has polarity. It consists of tiers of cells. As in the frog blastula, the top is the animal pole and the bottom the vegetal pole. Different tiers of blastula cells are separated, they show different developmental potential. Only cells from the vegetal pole are capable of initiating the development of a complete larva. It has been proposed that the reason for these differences is an uneven distribution of various transcriptional regulatory proteins in the egg cytoplasm. As cleavage progresses, these proteins end up in different combinations in different groups of cells. Therefore, specific sets of genes are activated in different cells, determining their different developmental capacities. Let's turn now to gastrulation in the frog, in which a number of key signaling molecules have been identified. Gastrulation in the frog begins at the great crescent. Amphibian blastulas have considerable yolk and are more than 1 cell thick. Therefore, gastrulation is more complex in amphibians than in sea urchins. Furthermore, there is considerable variation among different species of amphibians. In this brief account, we will mix results from studies done on different species to produce a generalized picture of amphibian development. Amphibian gastrulation begins when certain cells in the great crescent change their shape and their cell adhesion properties. The main bodies of these cells bulge inward toward the blastocele while they remain attached to the outer surface of the blastula by slender necks. Because of their shape, these cells are called bottle cells. The bottle cells mark the spot where the dorsal lip of the blastopore will form figure 11. As the bottle cells move inward, they create this lip, over which successive sheets of cells will move into the blastocele in a process called involution. The first involuting cells are those of the prospective endoderm, and they form the primitive gut, or archenteron. Closely following are the cells that will form the mesoderm. As gastrulation proceeds, cells from the animal hemisphere move toward the site of involution in a process called epiboly. Blastopore lip widens and eventually forms a complete circle surrounding a plug of yolk-rich cells. As cells continue to move inward through the blastopore, the archenteron grows, gradually displacing the blastocele. Institute for Advanced Studies, Jodhpur 25. As gastrulation comes to an end, the amphibian embryo consists of 3 germ layers, ectoderm on the outside, endoderm on the inside, and mesoderm in the middle. The embryo also has a dorsal ventral and anterior posterior organization. Most importantly, however, the fates of specific regions of the endoderm, mesoderm, and ectoderm have been determined. The discovery of the events whereby determination takes place in the amphibian embryo is one of the most exciting stories in animal development. The dorsal lip of the blastopore organizes embryo formation. In the 1920s, the German biologist Hans Spemann was studying the development of salamander eggs. He was interested in finding out whether the nuclei of blastomeres remained totipotent, capable of directing the development of a complete embryo. With great patience and dexterity, he formed loops from a single human baby hair to constrict fertilized eggs, effectively dividing them in half. Spemann loops bisected the great crescent, both halves of the zygote gastrulated and developed into complete embryos, experiment 1 and figure 12. But when the great crescent was on only one side of the constriction, only the half of the zygote developed into a complete embryo. The half lacking great crescent material became a clump of undifferentiated cells that Spemann called the belly piece, experiment 2 and figure 13. Spemann thus hypothesized that cytoplasmic determinants in the region of the great crescent are necessary for gastrulation and thus for the development of a normal organism. To test his hypothesis, Spemann and his student Hild Mangold conducted a series of delicate tissue transplantation experiments. They transplanted pieces of early gastrulus to various locations on other gastrulas. Guided by fate maps, see figure 8, they were able to take a piece of ectoderm they knew would develop into skin and transplant it to a region that normally becomes part of the nervous system, and vice versa. They performed these transplants in early gastrulas. The transplanted pieces always developed into tissues that were appropriate for the location where they were placed. Donor epidermis, that is, cells destined to become skin in their original location, developed into host neural ectoderm, nervous system tissue, and donor presumptive neural ectoderm developed into host skin. Thus, the fates of the transplanted cells had not been determined before the transplantation. In late gastrulas, however, the same experiment yielded opposite results. Donor presumptive epidermis produced patches of skin cells in the host nervous system, and donor presumptive neural ectoderm produced nervous system tissue in the host skin. Something had occurred during gastrulation to determine the fates of the embryonic cells. In other words, as Spemann and Mangold had hypothesized, the path of differentiation a cell would follow was determined during gastrulation. Spemann next did an experiment that produced momentous results. They transplanted the dorsal lip of the blastopore, figure 13. A small piece of tissue was transplanted into the presumptive belly area of another gastrula. It stimulated a second site of gastrulation, and second whole embryo formed belly to belly with the original embryo. Because the dorsal lip of the blastopore was apparently capable of inducing the formation of an entire embryo, Spemann and Mangold dubbed it the primary embryonic organizer, or simply the organizer. Molecular mechanisms of the Organizer. In recent years, researchers have studied the primary embryonic organizer intensively to discover the molecular mechanisms involved in its action. The transcription factor beta-catenin in the late blastula corresponds to the location of the organizer in the early gastrula, so beta-catenin is a candidate for the initiator of organizer activity. To prove that a protein is an inductive signal, it has to be shown that it is both. Institute for Advanced Studies, Jodhpur 26. Necessary and sufficient for the proposed effect. In other words, the effect should not occur if the candidate protein is not present necessity, and the candidate protein should be capable of inducing the effect where it would otherwise not occur sufficiency. The criteria of necessity and sufficiency have indeed been satisfied for the transcription factor beta-catenin. If beta-catenin mRNA transcripts are depleted by injections of antisense RNA into the egg, gastrulation does not occur. Beta-catenin is experimentally overexpressed in another region of the blastula. It can induce a second axis of embryo formation, as the transplanted dorsal lip did in the Spemann Mangold experiments. Beta-catenin appears to be both necessary and sufficient for the formation of the primary embryonic organizer, but it is only one component of a complex signaling process. What follows is a summary of some of the critical early steps in this signaling cascade. This description may contain a confusing amount of detail. However, it is not the arcane names of the genes and gene products involved that are important to remember. Third, we hope to provide a basic understanding of how these signaling molecules, their interactions and their gradients, can create invade positional and temporal information. Studies of early gastrulas reveal that primary embryonic organizer activity is induced by signals emanating from vegetal cells just below the gray crescent. Protein beta-catenin appears to play critical roles in generating these signals. One signal critical to stimulating the expression of organizer genes is the transcription factor glucocoid. Expression of the glucocoid gene appears to depend on two signaling pathways, both of which involve beta-catenin. The first of these pathways involves a glucocoid-promoting transcription factor called CMA. Is normally repressed by a ubiquitous transcription factor called TCF3, but in cells where beta-catenin is present, an interaction between TCF3 and beta-catenin induces CMA expression, figure 14. But CMA protein alone is not sufficient for glucocoid expression. Cells receive mRNA transcripts from the original egg cytoplasm for proteins in the TGF-beta, transforming growth factor beta, superfamily of cell signaling molecules. One or more of these genes candidates include VG1 and nodal interact with CMA protein by cooperatively binding to the promoter of the glucocoid gene and thereby controlling its transcription, see figure 14. Thus it is a particular combination of factors that determine which cells become the primary organizer. Cells that receive other combinations of signaling molecules are induced to become different types of mesoderm. Molecular mechanisms of left-right axis formation. We have seen how the distribution of cytoplasmic determinants in the egg can set up a dorsal ventral axis, and how the site of sperm entry can set up an anterior-posterior axis. What about the left-right body axis? After all, not everything in the animal is bilaterally symmetrical. The internal organs of a vertebrate have many left-right asymmetries. In humans, the heart is tilted to the right side of the body, the aorta comes off of the left side of the heart and the pulmonary artery comes off of the right side of the heart. The spleen is on the left side of the body, and the large intestine goes from right to left, to name only a few. We now know that there are a number of genes that are necessary for normal left-right organization of the body. If one of these genes is knocked out, it can randomize the left-right organization of the internal organs, with serious, even lethal, consequences. What triggers the asymmetrical expression of these genes? We do not know the complete answer to this question, but it appears that the mechanism involves a left-right differential distribution of some of the transcription factors that act very early during gastrulation. For example, in frogs, one of the TGF-beta proteins involved in organizer determination is also responsible for determining the left-right axis. In mammals, there are cilia that cause a differential flow of fluid in the yolk sac cavity. If these cilia are inactivated, the normal left-right asymmetries of the internal organs become random. Reptilian and avian gastrulation is an adaptation to yolky eggs. The eggs of reptiles and birds contain a massive yolk, and therefore the blastulas of these species develop as a disc of cells on top of the yolk, see figure 6c. We will use the chicken egg to show how gastrulation proceeds in a flat disc of cells rather than in a ball of cells. Cleavage in the chick results in a flat, circular layer of cells called a Institute for Advanced Studies, Jodhpur 27. Blastodisc. Between the blastodisc and the yolk mass is a fluid-filled space. Some cells from the blastodisc break free and move into this space. Other cells grow into this space from the posterior margin of the blastodisc. These cells come together to form a continuous layer called the hypoblast, which will later give rise to extraembryonic membranes that will support and nourish the developing embryo. The overlying cells make up the epiblast, which will form the embryo proper. Thus, the avian blastula is a flattened structure consisting of an upper epiblast and a lower hypoblast, which are joined at the margins of the blastodisc. The blastocele is the fluid-filled space between the epiblast and hypoblast. Gastrulation begins with a thickening in the posterior region of the epiblast caused by the movement of cells toward the midline and then forward along the midline, figure 15. The result is a midline ridge called the primitive streak. A depression called the primitive groove forms along the length of the primitive streak. The primitive groove functions as the blastopore, and cells migrate through it into the blastocele to become endoderm and mesoderm. In the chick embryo, no archenderm forms, but the endoderm and mesoderm migrate forward to form the gut and other structures. At the anterior end of the primitive groove is a thickening called Henson's node, which is the equivalent of the dorsal lip of the amphibian blastopore. In fact, many signaling molecules that have been identified in the frog organizer are also expressed in Henson's node. Cells that are Henson's node become determined by the time they reach their final destination, where they differentiate into certain tissues and structures of the head and dorsal midline, but not the nervous system. Mammals have no yolk, but retain the avian reptilian gastrulation pattern. Mammals and birds both evolved from reptilian ancestors, so it is not surprising that they share patterns of early development, even though the eggs of mammals have no yolk. Earlier we described the development of the mammalian trophoblast in the inner cell mass, which is the equivalent of the avian epiblast. As in avian development, the inner cell mass splits into an upper layer called the epiblast and a lower layer called the hypoblast, with a fluid-filled cavity between them. The embryo will form from the epiblast, and the hypoblast will contribute to the extraembryonic membranes, figure 16. The epiblast also contributes to the extraembryonic membranes, specifically, it splits off an upper layer of Institute for Advanced Studies, Jodhpur 28. Cells that will form the amnion. Cells that will form the amnion. The amnion will grow to surround the developing embryo as a membranous sac filled with amniotic fluid. Gastrulation occurs in the mammalian epiblast just as it does in the avian epiblast. A primitive groove forms, and epiblast cells migrate through the groove to become layers of endoderm and mesoderm. Neurulation, initiating the nervous system. Gastrulation produces an embryo with 3 germ layers that are positioned to influence one another through inductive interactions. During the next phase of development, called organogenesis, many organs and organ systems develop simultaneously and in coordination with one another. The process of organogenesis that is directly related to gastrulation is neurulation, the initiation of the nervous system in vertebrates. We will examine this event in the amphibian embryo, but it occurs in a similar fashion in reptiles, birds, and mammals. The stage is set by the dorsal lip of the blastopore. The first cells to pass over the dorsal lip of the blastopore move anteriorly and become the endodermal lining of the digestive tract. Following these first cells over the dorsal lip are those that will become mesoderm, see figure 11. The dorsal mesoderm closest to the midline, the shorter mesoderm, will become a rod of connective tissue called the notochord. The notochord gives structural support to the developing embryo. It is eventually replaced by the vertebral column. After neurulation, the shorter mesoderm induces the overlying ectoderm to begin forming the nervous system. Neurulation involves the formation of an internal neural tube from an external sheet of cells. First signs of neurulation are flattening and thickening of the ectoderm overlying the notochord. This thickened area forms the neural plate, figure 17. The edges of the neural plate that run in an anterior-posterior direction continue to thicken to form ridges or folds. In these neural folds, a groove forms and deepens as the folds roll over it to converge on the midline. The folds fuse, forming both a cylinder, the neural tube, and a continuous overlying layer of epidermal ectoderm. The neural tube develops bulges at the anterior end, which become the major divisions of the brain. The rest of the tube becomes the spinal cord. In humans, failure of the neural tube to develop normally can result in serious birth defects. If the neural folds fail to fuse in a posterior region, the result is a condition known as spina bifida. If they fail to fuse at the anterior end, an infant can develop without a forebrain, a condition called anencephaly. Whereas several genetic factors that can cause neural tube defects have been identified, there are also environmental factors, including dietary ones. The neural tube defects used to be about 1 in 300 live births, but we now know that this incidence can be cut in half if pregnant women have an adequate amount of folic acid AB vitamin in their diets. Institute for Advanced Studies, Jodhpur 29. Body segmentation develops during neurulation. Like the fruit flies whose development, vertebrates have a body plan consisting of repeating segments that are modified during development. These segments are most evident as the repeating patterns of vertebrae, ribs, nerves, and muscles along the anterior-posterior axis. As neural tube forms, mesodermal tissues gather along the sides of the notochord to form separate blocks of cells called somites, figure 18. The somites produce cells that will become the vertebrae, ribs, and muscles of the trunk and limbs. The nerves that connect the brain and spinal cord with tissues and organs throughout the body are also arranged segmentally. The somites help guide the organization of these peripheral nerves, but the nerves are not of mesodermal origin. When the neural tube fuses, cells adjacent to the line of closure break loose and migrate inward between the epidermis and the somites and under the somites. These cells, called neural crest cells, give rise to a number of structures, including the peripheral nerves, which grow out to the body tissues and back into the spinal cord. Development progresses, the segments of the body become different. The spinal cord differ. Regions of the vertebral column differ in that some vertebrae grow ribs of various sizes and others do not. Forelegs arise in the anterior part of the embryo, and hind legs arise in the posterior. Hox genes control development along the anterior-posterior axis. Homeobox genes are central to the process of anterior-posterior determination and differentiation. Earlier, we saw how homeotic genes control body segmentation in Drosophila. In the mouse, 4 families of homeobox genes, called Hox genes, control differentiation along the anterior-posterior body axis. The mammalian Hox gene family resides on a different chromosome and consists of about 10 genes. Remarkable is that the temporal and spatial expression of these genes follows the same pattern as their linear order on their chromosome. That is, the Hox genes closest to the 3' end of each gene complex are expressed first and are expressed in the anterior of the embryo. The Hox genes closer to the 5' end of the gene complex are expressed later and in a more posterior part of the embryo. As a result, different segments of the embryo receive different combinations of Hox gene products, which serve as transcription factors, figure 19. What causes the linear, sequential expression of Hox genes is unclear. Hox genes give cells information about their position on the anterior-posterior body axis, other genes give cells information about their dorsal-ventral position. Institute for Advanced Studies, Jodhpur 30. Tissues in each segment of the body differentiate according to their dorsal-ventral location. In the spinal cord, for example, sensory nerve connections develop in the dorsal region and motor nerve connections develop in the ventral region. In somites, dorsal cells develop into skin and muscle and ventral cells develop into cartilage and bone, see figure 18. An example of a gene that provides dorsal-ventral information in vertebrates is sonic hedgehog, which is expressed in the mammalian notochord and induces cells in the overlying neural tube to have fates characteristic of ventral spinal cord cells. As with the Hox genes, sonic hedgehog is homologous to a Drosophila gene, which is known simply as hedgehog. One family of homeobox genes, the PAX genes, plays many roles in nervous system and somite development. One of the genes, PAX3, is expressed in those neural tube cells that will develop into dorsal spinal cord structures. Sonic hedgehog represses the expression of PAX3, and their interaction is one source of dorsal-ventral information for the differentiation of the spinal cord. For the development of body segmentation, the formation of organs and organ systems progresses rapidly. The development of an organ involves extensive inductive interactions of the kind we saw in the example of the vertebrate eye. These inductive interactions are a current focus of study for developmental biologists. Extraembryonic membranes. There is more to a developing reptile, bird, or mammal than the embryo itself. The embryos of these vertebrates are surrounded by several extraembryonic membranes, which originate from the embryo but are not part of it. The extraembryonic membranes function in nutrition, gas exchange, and waste removal. Extraembryonic membranes form with contributions from all germ layers. We will use the chicken to demonstrate how the extraembryonic membranes form from the germ layers created during gastrulation. The yolk sac is the first extraembryonic membrane to form, and it does so by extension of the endodermal tissue of the hypoblast layer along with some adjacent mesoderm. The yolk sac grows to encloses the entire body of yolk in the egg. It constricts at the top to create a tube that is continuous with the gut of the embryo. However, yolk does not pass through this tube. digested by the endodermal cells of the yolk sac, and the nutrients are then transported to the embryo through blood vessels that form from the mesoderm and line the outer surface of the yolk sac. The allantoic membrane is also an outgrowth of the extraembryonic endoderm plus adjacent mesoderm. It forms the allantois, a sac for storage of metabolic wastes. Just as the mesoderm of the hypoblast grow out from the embryo to form the yolk sac and the allantoic membrane, ectoderm and mesoderm combine and extend beyond the limits of the embryo to form the other extraembryonic membranes. Two layers of cells extend all along the inside of the eggshell, both over the embryo and below the yolk sac. Where they meet, they fuse, forming two membranes, the inner amnion and the outer chorion. The amnion surrounds the embryo, forming the amniotic cavity. The amnion secretes fluid into the cavity, providing a protective environment for the embryo. The outer membrane, the chorion, forms a continuous membrane just under the eggshell, figure 20. It emits water loss from the egg and also works with the enlarged allantoic membrane to exchange respiratory gases between the embryo and the outside world. Extraembryonic membranes in mammals form the placenta. In mammals, the first extraembryonic membrane to form is the trophoblast, which is already apparent by the fifth cell division, see figure 7. When the blastocyst reaches the uterus and hatches from its encapsulating zona pellucida, the trophoblast cells interact directly with the endometrium. Adhesion molecules expressed on the surfaces of these cells attach them to the uterine wall. By excreting proteolytic enzymes, the trophoblast burrows into the endometrium, beginning the process of implantation, see figure 16. Eventually, the entire trophoblast is within the wall of the uterus. The trophoblastic cells then send out numerous projections, or villi, to increase the surface area of contact with maternal blood. Meanwhile, the hypoblast cells extend to form what in the bird would be the yolk sac. But there is no yolk in mammalian eggs, so the yolk sac contributes mesodermal. Institute for Advanced Studies, Jodhpur 31. Tissues that interact with trophoblast tissues to form the chorion. The chorion, along with tissues of the uterine wall, produces the placenta, the organ of nutrient, respiratory gas, and metabolic waste exchange between the mother and the embryo, figure 21. In time the yolk sac is forming from the hypoblast, the epiblast produces the amnion, which grows to enclose the entire embryo in a fluid-filled amniotic cavity. The rupture of the amnion and chorion and the loss of the amniotic fluid, water breaking, herald the onset of labor in humans. An allantois also develops in mammals, but its importance depends on how well nitrogenous wastes can be transferred across the placenta. In humans the allantois is minor, in pigs it is important. In humans and other mammals, allantoic tissues contribute to the formation of the umbilical cord, by which the embryo is attached to the chorionic placenta. It is through the blood vessels of the umbilical cord that nutrients and oxygen from the mother reach the developing fetus and wastes, including carbon dioxide and urea, are removed, see figure 21. The extraembryonic membranes provide means of detecting genetic diseases. Cells slough off of the developing human embryo and float in the amniotic fluid that bathes it. Later in development, a small sample of the amniotic fluid may be sampled with a needle as the first step of a process called amniocentesis. Cells of fluid can be cultured and used for biochemical and genetic analyses that can reveal the sex of the fetus, as well as genetic markers for diseases such as cystic fibrosis, Tay-Sachs disease, and Down syndrome. If amniocentesis is performed, it is usually not until after the 14th week of pregnancy, and the tests require 2 weeks to complete. If abnormalities in the fetus are detected, termination of the pregnancy at that stage would put the mother's health at greater risk than would an earlier abortion. Therefore, a newer technique, called chorionic villus sampling, is now in common use. In this test, a small sample of the tissue from the surface of the chorion is taken, figure 22. This test can be done as early as the 8th week of pregnancy, and the results are available in several days. Human development. In humans, gestation, or pregnancy, lasts about 266 days, or 9 months. Our mammal station is shorter, for example, 21 days in mice, and in larger mammals it is longer, for example, 330 days in horses and 600 days in elephants. The of human gestation can be divided into 3 periods of roughly 3 months each, called trimesters. Intrauterine development can be divided into 3 trimesters. The first trimester, implantation of the human blastocyst begins on about the 6th day after fertilization. After implantation, gastrulation occurs, the placenta forms, tissues differentiate, and organs begin to develop. The heart begins to beat in week 4, and limbs form by week 8, figure 23a. Most organs have started to form by the end of the first trimester. By that time, the embryo looks like a miniature version of the adult, and is called a fetus. The first trimester is a time of rapid cell division and tissue differentiation. Signal transduction cascades and the resulting branching sequences of developmental processes are in their early stages. Therefore, the first trimester is the period during which the embryo is most sensitive to damage from radiation, drugs, chemicals, and pathogens that can cause birth defects. An embryo can be damaged before the mother even knows she is pregnant. Institute for Advanced Studies, Jodhpur 32. Classic and tragic case is that of thalidomide, a drug widely prescribed in Europe in the late 1950s to treat nausea. Women who took this drug in the 4th and 5th week of pregnancy, when the embryo's limbs are beginning to form, gave birth to children with severely malformed arms and legs. This cause major and noticeable responses in the mother during the first trimester, even though the fetus at the end of that time is still so small that it would fit into a teaspoon. Soon after the blastocyst implants itself, it begins to secrete human chorionic gonadotropin, HCG. This hormone stimulates the mother's ovary to continue to produce the hormones estrogen and progesterone, which help to maintain the pregnancy. These hormonal changes cause the well-known symptoms of pregnancy: morning sickness, mood swings, changes in the senses of taste and smell, and swelling of the breasts. During the second trimester the fetus grows rapidly to a weight of about 600 grams, and the mother's abdomen enlarges considerably. The limbs of the fetus elongate, and the fingers, toes, and facial features become well-formed, figure 23b. The movements are first felt by the mother early in the second trimester, and they become progressively stronger and more coordinated. By the end of the second trimester, the fetus may suck its thumb. The third trimester, the fetus and the mother continue to grow rapidly during the third trimester. Though the embryo is most susceptible to adverse effects of drugs, chemicals, and diseases during the first trimester, the potential for serious effects of exposure to many environmental factors continues throughout pregnancy. Protein malnutrition, alcohol consumption, and cigarette smoking are examples of factors that can result in low birth weight, mental retardation, and other developmental complications. As the third trimester approaches its end, many internal organs mature. The digestive system begins to function, the liver stores glycogen, the kidneys produce urine, and the brain undergoes cycles of sleep and waking. Developmental changes continue throughout life. Development does not end with birth. Obviously, growth continues until adult size is reached, and even when growth stops, organs of the body continue to repair and renew themselves through cycles of cell replacement by the progeny of undifferentiated stem cells.